Hello, everyone. Today we're going to have a lecture for immune system. This is going to be the last lecture for Unit 3. Um, I'm going to start with do a little review on the non-specific immune responses. Let me get my screen up here. Okay, so at the end of last lecture, I told you that you need to read this part, the non-specific immune defenses system. If you have not done that, please stop and go back to read your book first. It's going to be on page 781 to 790. You really have to read this part first and then come back to watch this video. Okay, so when we talk about immune defense systems, there are two big arms. One is called non-specific system. Another one is called a specific defense system. And in your book on page 782, there is a figure 21.1. It gave you an idea. Okay, non-specific system is also called innate defenses. The specific defense system is also called adaptive defenses. Okay, so if it's a non-specific or innate defenses, there are two parts. The first one is the called first line of defense, which is the body surface. For example, your skin covers the whole body and to prevent antigens, foreign antigens to get in to your internal systems unless you have a cut. So that is pretty much the first line of defense. And also, if you can think, there are some sweat glands, the hair, sweat gland will secrete acidic fluid, and that will inhibit the bacteria growth. So all these are considered as first line of defense. The second line of defense, it is still, please remember, it is still non-specific defense mechanisms. Okay, so let me go over these second line of defense mechanisms. Okay, the first one is by phagocytosis. We do have a phagocytes like macrophages, dendritic cells, also specific macrophages stay in specific locations in our bodies. They don't move a lot to other parts of the bodies. Like this one is called Kupffer cells. This is really just the macrophage-like cells in the liver. Okay, neutrophils, they are white blood cells. They are phagocytes. Eosinophils, they are phagocytes, but very weak ones, And another one is B cells, B lymphocytes. All these are considered as phagocytes. Okay, so the mechanisms for phagocytosis is on page 784. So if you need to review, please go back and follow the chart and write out the step-by-step process for phagocytosis. That's this chart. Okay, the next one is called natural killer cells. All right, natural killer cells can work to defend our bodies. This mechanism is belong to the second line of defense. So this chart kind of give you an idea, okay? If the natural killer cells sense there is a abnormal cells in your body, like infected cells or cancerous cells, any abnormal cells, all right? And then these natural killer cells will going to move toward to these abnormal cells and then form a connection right here. If you enlarge, it looks like this. Okay, this side is natural killer cell, this side is abnormal cell. So right here, when this natural killer cell is very close to this abnormal cells, it's going to start releasing a chemical protein. It's called a perforin. Perforin. This chemical, this substance is going to form a pores on this abnormal cells. So kind of make a pinched holes on these abnormal cells and then destroy this cell. So this mechanism is called the natural killer cells mechanism. And by releasing perforins to destroy abnormal cells. This one is not specific. As long as the natural killer cells sense there's abnormal cells around, it's going to trigger this response. Okay, the third mechanism for second line of defense is called inflammation. This process is listed in your book on page 786. So there are 4 cardinal signs associated with inflammation. They are redness, heat, swelling, and pain. Redness because the blood vessels become more permeable. There will be more blood in that area, so it's more red. And heat is because blood is always warmer than the rest of the body. So if you have more blood to that area, then it's going to be warmer. So if you have a cut, you have inflammation on your hand, if you touch it, it's kind of warm. We all know that, okay? The third one is swelling because the blood vessels are more permeable, which will allow more fluid coming out from blood. And then so you're going to have edema around this trauma-sized area. That's why you have a swelling tissue. The pain is because the extra fluid coming out from blood will compress the nerve endings around the area and cause pain. So that's the 4 cardinal signs. Okay, so this process is listed in on page 786. Please review this. Okay, let me just mention right here. Okay, so once you have the chemicals released from the damaged tissue, which will trigger the neutrophils and other phagocytes to come to this damaged site. So the neutrophils will and other phagocytes like macrophages, they're traveling in the blood capillaries, then they get attached to the wall of the capillaries, and then they can sneak out. This sneaking out process is called diapedesis. Okay, once the phagocytes get to this side, it can start to destroy or take away these abnormal particles. Okay, so this is a review slide for inflammatory responses. Okay, next mechanism, it is still the second line of defense, is called antimicrobial proteins. All right, there are 2 types, 2 big types. One is called interferons, you may heard about. Another one is called complement proteins. Okay, so let's talk about interferons first. Interferons work in a unique way because interferons, they can only protect uninfected cells. Okay, let me share my whiteboard with you here. Let's see. Okay. Sorry, give me a second. Okay. All right, everyone. So interferons like this. So let's say if this is a liver cell, okay, and then you had infection, okay, so let's say viral infection in here. So this is a infected cell. Then this cell, because of the infection, will trigger this cell to release interferons. Okay, interferons are type of proteins. There are many subtypes of interferons. So it's just this type of proteins called interferons. So this interferons, what can they do? They can move toward to the surrounding cells. These cells are not being infected yet. So this interferon now can bind to the receptors here, and receptors here. Once this binding takes place, it's going to make these cells very resistant to this infectious agent. For in this example, is going to be the viruses. Okay, so the interferons can only protect the non-infected cells. For this one, is already infected, then you cannot do anything. The interferon cannot help this one, cannot help cell A, but it can help cell B, C, and D because they're non-infected. By having interferon released from cell A, it can bind to the receptor on the surrounding cells and make the surrounding cells more resistant to the viruses around. That's how interferon works. Okay, let's back to this slide. Okay, now next one is, let me go back. Okay, interferon. So please remember, interferons work to protect non-infected cells. All right, complement proteins. There are about 20 of different complement proteins. They're all circulating in the blood plasma. Normally, they're in the inactive forms unless there's a trigger factor to trigger the activation process. Okay, so once this process is triggered, then this complement proteins, they're going to work like the coagulant factors, kind of like a one activation lead to the next activation, and it's a cascade activation process, and eventually to form a structure called membrane attack complex, MAC. Everyone, please write it down. Okay, membrane attack. Let me go back. Membrane attack complex, MAC. This MAC formation will destroy the abnormal cells or the infected cells. Let me give you the diagram here. Okay, so there are two different pathways. Okay, classical pathway, alternative pathway. You have these in your book. You don't need to remember the specific complement proteins involved in each step, but you need to know both pathways. Eventually, they're going to lead to the activation of C3, complement protein number 3. Once the C3 is activated, it's going to go this in the middle path. Is going to form this MAC. What is MAC? Membrane attack complex. This is kind of like a pinched holes on the surface of bacteria. You see that? Because you form these pinched holes, there's so many of them, and you totally destroy the bacteria's cell membrane, and then you destroy the bacteria. So this is one major way. Okay, and at the same time, once you activate C3, you can activate a process. It's called opsonization. Okay, that is, you put something on the surface of this bacteria, which will allow your phagocytes to recognize this bacteria much more easier. So I say, oh, okay, there's something here. It's labeled as abnormal stuff. We're going to start the immune responses to destroy them. Okay, and also at the same time, once you have C3 activated, it can trigger the inflammation. We already talked about inflammation, right? So the complement proteins work with two different pathways, and once they activate C3 protein, and then you have three major ways to to trigger these immune responses. One is by form MAC, one is by labeling the bacteria, and then one is by causing inflammation. All right, the fever is another mechanism for the second line of defense. Okay, so fever is caused by the activation of pyrogens, and also by a cytokine protein. It's a protein. It's called interleukin-1. This one is released by macrophages. Once you have infections, the macrophages can release interleukin-1. So pyrogens is the protein which can increase, which can sense your hypothalamus to increase the body temperature. Okay. Just think about a fever, it is not all bad. Fever can actually be beneficial to you, because fever, higher temperature, most of bacteria cannot tolerate higher temperatures, so they cannot multiply fast enough so that will give you more time to destroy them. And also okay, this part, let me just say, you read your book, it's pretty straightforward. Okay, so fever, as long as it's not too high, it should be beneficial to you. So next time, if you have a fever, try to lower body temperature with a physical methods, like take a bath or have alcohol wiping, but wait for Tylenols. If you have like a very high fever, of course you want to have medicines. Otherwise, you may want to just let it down by itself, so the fever can help you to inhibit the bacteria growth and give you more time to fight for them. Okay, so this is the summary page for all the non-specific defense mechanism. The first one is the physical barriers. That is the first line of defense. The rest of them are the second line of defense mechanisms. So please remember, all of these are non-specific defense mechanisms. Okay, that's the summary for today. And then we're going to focus on the next topic, which is called adaptive or specific defense mechanisms. Before we start, we need to have some, I need to give you some terms. Okay, so first is antigens. We already talked about antigens on the previous lecture, like a blood type antigens. That is some substance which can trigger the immune responses. Okay, so antigens, a lot of them are proteins, but they can be sugar, they can be like particles, they can be pollen. So anything that can trigger your immune responses can be called antigen. So if you look at an antigen, a lot of times it's not this whole cell cause problem. It's the molecule specific parts on the surface of this cell can cause immune responses. So these specific parts on the cell or the molecules on the cell is called antigenic determinants. Antigenic determinants. So in this case, you can see this is an antigen. Let's say this is a bacteria. bacterium. And on this bacterium surface, you have different antigenic determinants, like different shape ones. So different shape, shape, shape. And then in your body, when you make antibodies, these antibodies are not used to against this whole antigen. Rather, they're very specific to each type of the shape here on the surface. Or we can say these antibodies are very specific for each type of this antigenic determinant. All right, so the cells involved in the specific immune responses. First, the T cells, T lymphocytes. We have two cells, okay? One is called T helper cells. It is also called CD4 cells. Another type is called T killer cells, or can be called T cytotoxic cells, or CD8 cells. The third one is the suppressors T cells. This one is used to balance the activities. So the most important ones are the T helper and T killer cells. And then the B lymphocytes, the B cells, we did mention it last time. When the B cells are activated, it's going to differentiate to become plasma cells. The main function for B cells or for the plasma cells is to produce antibodies. Okay, so remember we talked about the proteins. Most of the proteins are made in the liver, but antibodies are all made in the plasma cells. Okay, next one is a macrophage. Okay, macrophage is called antigen presenting cell. Okay, that's the type of cells. It's called antigen presenting cell. I will explain this. APC, antigen presenting cells. So besides of macrophages, we have dendritic cells and B lymphocytes. They're all antigen presenting cells. So these cells, they have a ability to present the bad or the foreign antigens on their surface. That's why it's called antigen presenting cells. So macrophages, dendritic cells, and B cells are the best APCs. Okay, this one is showing you the dendritic cell. Okay, so a little bit background about B cells and T cells. Okay, so B cells and T cells, they circulate in your blood. Let's just use T cells as example. So T cells are matured in the thymus. All right, so we made this selection process. The selection process takes place in the thymus. And once you have these B cells made, they're going to be circulating in the blood. So all of these T cells, they are very unique. On each one of these T cell surface, they carry a specific receptor which can recognize a specific antigenic determinant. Okay, so just remember now, T cells and B cells, they are all, well, let's just use T cells as the example. T cells, among all these T cells, you may have 1 trillion or more than 1 trillion of them, and all of them are different in terms of the receptor on each one of them. Okay, so each receptor can only recognize a very specific antigen. All right, and next term is called MHC proteins. Okay, this one is a major histocompatibility complex, MHC. Okay, So in our cells, we have two types of MHC. One is class 1 MHC or MHC1. This one, please remember, you can find in all of your body cells except RBCs. Another word, we can find these MHC1 proteins on all the nucleated body cells. Okay, RBCs, the mature ones, do not have nuclei. So we say MHC1 can be found in all nucleated cells. Okay, on the cell membrane. MHC2 is the proteins you can only find on APCs. What is APC? Antigen presenting cells. What type of cells are APCs? Macrophages, dendritic cells, and B cells. Okay, one more time. Where can you find MHC1? All body cells except RBCs. Where can you find MHC2? Only on antigen presenting cells. Okay. All right, so now this one is talking about the structure of MHC proteins. The structure of MHC proteins. Okay, so they say each MHC protein molecule has a deep groove which can carry a little peptide. What is a peptide? If you digest a big protein to fragments, and each little fragment is called peptides, right? So this peptide is going to be stuck in this groove of MHC protein or molecule, and then transported to the surface of the cell membrane. Okay, so this one give you a MHC protein production process. Okay, so this one definitely for MHC1 molecules. All right, MHC1, where can you find it? In what type of cells? All of your body cells except RBC, right? So this MHC protein is made in the rough ER. Okay, so the structure looks like this. There is a groove right here. Okay, so this groove will get a little peptide stuck on it. Then it becomes a functional MHC1 protein, and then can be transported to the surface. So now you see this is an MHC1 with a little peptide on it. Okay, that is the whole structure of MHC1. The important part is about this peptide here. This peptide is called endogenous peptide. Endogenous peptide. Endogenous means this peptide is actually made by this cell. Oops, too fast. Okay, this peptide, if it's made in this cell, is called endogenous peptide. Let me give you an example. I say, we are in each of our liver cell, for example, in each of our liver cell, we constantly making new proteins, and also we digest proteins as well. Okay, so when you digest yourself proteins, you're going to make many of these self peptide fragments. Okay, these fragments are called endogenous peptides because the protein is originally made in this cell and is digested in this cell, and this peptide is your own peptide. You made that peptide before. So in this case, it is an endogenous peptide. So MHC1 can only bind to the endogenous peptide. So this whole thing here is an endogenous peptide. Okay, so this endogenous peptide can be normal, can be abnormal. So let's say if you digest a cancerous protein, then you're going to have a cancerous, you're going to have a peptide fragment from the cancerous protein. Then this peptide is going to be considered abnormal, right? Okay, so that's going to be on this MHC, okay? And if it's a normal cell, you have all the normal proteins, then on MHC1, you're going to have a normal peptide on the surface, all right? So that's MHC1. It only combined to the endogenous peptide, everyone. Keep that in mind. Okay, now MHC2, again, is made in the rough ER, okay? And then it's going to have a little peptide stuck on it, on the surface of it, and then transported to the surface of the cell. But this time, this peptide is not self-made. It is called exogenous antigen, exogenous peptide. So this one has to be a phagocyte, you see. This, let's say if this is a macrophage, okay, engulfing a bacterium in, and then lysozyme will destroy this bacteria. Once you destroy the bacterium, you're going to generate a lot of peptide fragments. A part of this fragment is going to get stuck on MHC2 and then travel to the surface. So now you see this macrophage is going to have MHC2 with a fragment from this bacterium. Okay, this is why we call this cell a antigen presenting cell, because this cell can present a piece of the bacteria fragment on the surface, and this one will trigger the immune responses. Okay, all right, let me repeat this one more time. Oops, too fast. Okay, if it's MHC1 molecules, it carries a endogenous peptide. If it's MHC2, it carries a exogenous peptide. All right, so where can you find MHC molecules? In what type of cells? Antigen presenting cells, including macrophages, dendritic cells, and B lymphocytes. Okay. All right, now we can talk about the actual mechanisms. There are two mechanisms, all right? One is called antibody-mediated response. This part is in your book on page 796. On 797, the bottom, the figure 21.13 is called humoral immunity. That is another word for antibody-mediated immunity. Okay, so this one is B lymphocytes involved immunity, specific immune responses. If it's a T lymphocytes involved immune responses, it's called cell-mediated specific immune responses. Okay, one more time, antigen presenting cells are the cells which have MHC2 on the surface. Okay, the good ones are macrophages, dendritic cells, and B lymphocytes. Okay, one more time. Antigen presenting cells, macrophage, dendritic cells, B cells. And what makes them unique? MHC2 on the cell surface. Alrighty, now we can look at the specific immune responses, the mechanisms. So I think I'm going to use my whiteboard for this part, and then we'll come back and review with the slides. Let me see, how do I do this? Here we go. Okay, so first one, we're going to talk about which one we should talk about first? The cell-mediated response. Okay, let's do the cell-mediated response first. Okay, the cell-mediated specific defense mechanism. Okay. Okay, so this is a T cell involved responses. So we talk about T cell, there's a T cell called a T killer cell. This one is also called a T cytotoxic cell, and also called CD8 cell. Yeah, you have to memorize that, okay? There's another type of cell is called T helper cell, and it's also called CD4 cell. Okay, and then we talked about, let me just review this. We talked about MHC1. Okay, where can you find it? All types of cells except RBC. Okay, MHC2 only on APCs. Are we good? Okay, then we can start. Okay, cell-mediated response. So it's more focused on the T cells. T cells, we have T killer, we have T helper. Okay. All right, so let's say if I have a liver cell infected or cancerous, let's say cancer liver cell. All right, and then on this surface, okay, you have a MHC1 molecule. This MHC1 molecule is going to have a little peptide stuck on it. Yes? Okay. So this MHC1 is can this be MHC2? No, because liver cell, okay, it's not a APC. So it only has MHC1. In this case, this MHC1 is carrying a abnormal endogenous peptide from liver, because this is a cancerous cell. All right, so the cancerous cell has abnormal peptides which get stuck on MHC1. All right, so in this case, in your blood circulation, you have many of the T cells, the T killer cells. Okay, remember I said you have more than 1 trillion of T killer cells. Each one has a unique receptor which can recognize this MHC1. So this one have a receptor here. This one has a different receptor. This one has another type of receptor. So out of these trillion T killer cells, only one of them, or let's say only a very few of them, have the same type of receptor which can only recognize this specific MHC1. When I say this specific MHC1, I meant this MHC1 carries a very specific peptide from this type of a cancer cell. All right, so this recognition process will take a while because you have trillion of these T killer cells. Only when this very specific one gets to this cancerous cell surface and recognize this MHC1, then it's going to trigger the specific cell-mediated response. What is that going to do? First, this T killer cell is going to release a protein, okay, interleukin. The different types of the cytokine. Let me just write down the cytokines. That's just the proteins. This cytokines is going to we call it co-stimulation. Co-stimulation. That means if this only when the T killer recognizes this MHC1, if it just recognizes it, bind to it, still cannot actually trigger this process unless this T killer start to release this cytokine proteins, which will help to trigger the immune responses. That's why it's called co-stimulation. So first, this T killer has to recognize this cancerous cells, MHC1, and then release a cytokine, and which will start the immune response. What it does is doing several things. Number one, it is going to release perforin. Do you remember this perforin? Natural killer cells can release perforin, which can, this perforin, if the perforin gets here, yeah, I can't do this. Perforin gets here, can make a holes on the surface of the cell and destroy the cell. That's one. Number two, it's going to trigger the T killer mitosis. Okay, so what is mitosis? It's going to make exactly the genetically identical T killer cells. So all of these cells made by this mitosis will have the same receptor on the surface. So basically, you're increasing the number of soldiers in the system try to go and find all the cancerous cells. Okay, so that's number two. But number two, this part, normally it's not fast enough unless you get some extra help. Okay, so it's kind of like if you're in a battle, okay, is that you're going to have more soldiers, you're going to have more enemies. These cancer cells, they can divide and make more and more so fast. They can spread it so fast. So the idea is you want to have enough T killers to get them all. Okay, how can you get enough T killers? You want to speed up the T killer cell mitosis so you can have more soldiers in the battle. So how can you get more soldiers? Okay, so this part, let me clear this one. This part requires T helper. Okay, T helper's job is to help to speed up the mitosis. Okay, so how can T helper work? Okay, this is how the T helper work. All right, T helper only recognize MHC2. Okay, so where can you find MHC2? On the APCs, right? So this one is a macrophage. All right, so on this one, on the surface, okay, you're going to have a MHC2 with a peptide on the surface. So this peptide, let me redo this. I did not get the right color. Okay, hold on one second. Okay, so this is a, oops, darn it. Okay, macrophage. All right, on the surface, you have a MHC2. Okay, so this one, you're going to have a peptide on it. All right, that is from cancerous cells. So let's just say if there's a cancer cell get engulfed in and then made this peptide. So this peptide is the same peptide as on the liver cell, but this one is not made by macrophage. It's engulfed in by macrophage. You see the difference? Okay, so if it's MHC2 molecule, it can only recognize T helper. Okay, T helper. T helper. MHC2 cannot recognize T killer, can only recognize T helper. Now again, all these T helpers, only one of them have the receptor which can recognize this specific MHC2. The rest of them may have different shape of receptors. They cannot react with this MHC2. So once this recognition takes place, then this T helper will release the cytokine. In this way, in this one is going to release something called interleukin 2. All right, so interleukin 2 will help to co-stimulate this process. Once this stimulation takes place, is the T helper. Okay, T helper is going to do the following things. So first, it's going to stimulate T helper mitosis to make more T helpers to help with this process. Okay, number two is going to help T killer cells to make more T killers by speed up mitosis. Okay, so once you have more T killers, then you can use all these T killers to in the circulation to take care of all the cancerous cells. You got it? Okay, so in this cell-mediated response, All right, you need to have T killer, you need to have T helper, you need to have APCs. All right, so the bottom line is you want to make a lot of this specific type of T killer cells. This is also called the CD8 cells. And then directly target on abnormal cells. Does that make sense? Okay. So once you have a lot of T killers, you have them going to the battle and destroy all the abnormal cells. At the same time, you're going to save some of them. Small amount of the T killers with this specific type of receptors will be saved for later, for the future. Okay, so next time, if you have the similar, if you have the same abnormal cells, then your system can recognize them much faster, okay, than the first time, because the first time you only have one cell in the whole circulation which can recognize this specific antigen, so it's going to take a while. Okay, but if you have some of them saved, then next time it's going to take less time to find them, and the response is going to be more dramatic. Okay, this is more true for the next part we'll talk about, the antibody mediated response. Okay, any questions you have on this part? Please write them down if you have questions. And this part of the lecture is it's on page 801 to 806. Okay, so stop here and go back to read the book one more time, and then come back. Okay, so this is a cell-mediated response. Okay, so now we can actually go back to, hold on, let me do this one first. Okay, let's go back to the slides. Okay, so we are talking about this side of the mechanism, activation by class 1 MHC protein. Okay, so we know on the surface of the cancerous cells, you're going to have MHC1 with a peptide, cancerous peptide, and this is a T killer cell. This is the cancerous cell, which can be recognized by T killer cell. T killer cell is also called CD8 cells, okay, and it's also called the cytotoxic T cells, which can directly destroy these abnormal cells by releasing perforin. Okay, at the same time, it's going to save some T cells as memory cells for the future. And remember, at the beginning we said the T cells, we also have a suppressor T cells, kind of to balance this process. You don't want to have too many of the T8, these cytotoxic T cells, too many of them, then it's just being wasted. So this one is just for balancing. Okay, so this is the first part of the first arm of the specific immune response is called cell-mediated immune responses. Let me just review this process one more time here. This is a abnormal cell here, or let's say it's a cancerous cell, all right? And then out of your circulation, only one of the T killer cell has the receptor which can recognize this, okay? This recognition process, if you enlarge this, okay, so on this side is MHC1 with a peptide. On this side is a T killer with a receptor. And on this side, not only the receptor needs to recognize this MHC1, but also there's another arm right here will help to secure this binding process. Okay, once this binding is secured, then this T killer cell can be activated and start mitosis to make the same T cells with the same receptors, and also save some as memory cells. But this process is not enough, is not fast enough unless you get help from the T helpers. So T helpers are on this side. These are T helper cells. Okay, so this is an APC. In this case, is a macrophage, which is going to have MHC2 on the surface. All right, T helper only recognize MHC2. And again, you have a side arm to secure this binding, and also release a cytokine to make this process to start. Okay, it's called co-stimulation. So once you activate this T helper, the T helper will go through mitosis and get more T helpers. And these T helpers are going to release the cytokines, which will stimulate the mitosis of T killer. Okay, so this is the T helpers. They can release interleukin 2, which can speed up mitosis for T killers. All right, so I think we're good. Okay, this is another review slide. Okay, so the T cell-mediated response, more targeting on the abnormal cells. When we say abnormal cells, definitely cancerous cells or infected cells or implanted foreign tissue. If you have an implanted foreign tissue, then your own T cells can recognize the MHC1 as abnormal and start this process. Okay, so you can use this slide as a review slide. Okay, so this one give you another review slide. Okay, so in this case, you see this is a macrophage, and this is a T killer cell, right? Okay, ooh, how can we have a T killer cell on the macrophage? T killer cell only binds to MHC1. Does macrophage has MHC1? Of course. All the nucleated cells have MHC1. Okay, and then this process, the whole point for having this binding is to start T killer cell mitosis to make a lot clones. Okay, same type of T killer cells. So, but this mitosis process, mitotic process, cannot be fast enough unless you have interleukin 2. So the interleukin 2 is the co-stimulate for the mitotic process. and this protein is released by activated T helper cells. So how can you activate T helper? T helper has to bind with MHC2. Okay, so please review this process. Okay, now the next topic is the B cell mediated response, also called antibody mediated response. And of course, this one is the B cells involved response. And we know when the B cell is activated, it's going to differentiate into plasma cells. Okay, and the plasma cells function is to make antibodies. So we want to have antibodies involved in this response. Okay, so this one I can just use this slide. I think this is good enough. All right, so these are the B cells we have in the circulation. Again, all of these B cells, they have different receptors on the surface, and each type of receptor only can recognize very specific antigen. Okay, so in this case, these antigens are these red dots, and there are 3 B cells. Only the middle one can recognize this antigen. Once this binding of antigen and receptor takes place, it's going to activate these B cells and start a mitotic process. Okay, so this mitotic process will make a lot of the same B cells, which will have the same receptors on the surface, and then they differentiate to make B cells, plasma cells. And all these plasma cells, the function is to make more of the same receptors. Okay, these receptors, you can also call them antibodies, or these antibodies, you can also call them receptors. Okay, so it is the protein, just like these receptors on the surface, but the function of them is they can bind with specific antigen and destroy them. Okay, so at the same time, just like T cells, some of these B cells with this type of receptors are going to be saved as memory cells for later. Okay, all right, so this is the first time, because you, out of the trillions B cells, you only have one cell which can recognize this specific antigen. So this process will take about a week, okay, to trigger this process, to trigger this mitotic process. So this is why when you get a flu, for example, it will take a week to get better, because it take a week to make these process to take place. Once you start to making these antibodies, and hopefully you're going to produce enough and then destroy all the antigens, and then you'll get better. Okay, and this memory cells is very, very important. It is on page 797. There is a chart to show you, once the first time of the response, if you look at the chart, antibody to A right there, it's around, okay, well, it's if you compare with the second part of the curve. So as long as you have the memories saved, next time if you are encountered with the same antigen, the response does not need to have a week to start, okay, because you already have the immunity, you already have the memory cells saved. So these cells can get into the battle right away. Okay, so the second time, the second response is, that's right here. The secondary response is much, much stronger and faster. This is the whole principle behind the vaccines. Okay, we want to give people vaccines. It's just like a very small dose of the antigen, either live or just the antigenic determinants, okay, to trigger your immune responses to make the memory cells. That's what we want. We want you to make the memory cells. So next time when you are encountered with the same antigen, then these cells can quickly be released into the battle, and then you can have much stronger immune responses, and then you will not get sick. Okay, you are going to win the battle because you have more soldiers. Okay, all right, so this is another review slide. Let's see, first is this B cells. All right, B cells with okay, first question, does B cell have MHC1? Does B cell have MHC2? The answer, yes. Okay, B cells have both MHC1 and MHC2. For MHC1, it's on all nucleated cells, right? For MHC2, it is the antigen presenting cell, right? So this one, B cell is antigen presenting cell. Okay, so if you have antigen bind to the surface receptor, and then it's going to activate B cells. All right, so the MHC2 can bind with the T helper. Remember, MHC2 can only bind to the T helper. Once you have a T helpers, it's going to release interleukin 2, which can speed up the mitotic process for B cells. Once the B cells, you have a lot of B cells, then you can differentiate them into the plasma cells, and then you're going to make antibodies. All these antibodies are going to be in your plasma, circulating in the blood to take care of all the free antigens. So for this part, the antibody structures you can skip. I'm not going to ask you to know the structure of antibodies, so we just skip this, okay? And then this is kind of give you an idea. Once the antigen and antibody bind together, how can this binding destroy the antigens? All right, so you can read this part from your book. Okay, this is another review slide. Okay, review slide. Okay. All right, this chart is important, and please read this chart and make sure you understand it. Okay, so active immunity, passive immunity. Okay, so active immunity has to be something trigger your own body to start produce antibodies. Passive is you get antibodies somehow. Okay. Review slide. Okay, let's look at the questions. Which lymphocyte is involved in the cell-mediated response? Cell-mediated has to be T cells. Memory cells are used to B as a boy. Transplant rejection reaction is caused by T cells. Function of the vaccine is to C, make B memory cells. Okay, that's it for this part of the lecture. Okay, I'll see you next time.